start out by talking about the biomaterials of Axigard. And so this product line is made from porcine small intestine submucosa. And again, it's an extracellular matrix. And the real benefit of this material is its porosity. It has this very open and porous structure. And the advantage of that porous structure is that the patient's own cells can enter the material and they can start laying down new matrix, which then basically remodels the material. It becomes the patient's new tissue layer. And I want to just say the picture on the bottom is through the edge of the tube, and that's where we have that porosity that allows the cells to get in. When you're looking at the surface of the tube, the porosity it's not porous like the edge is, and that's because we don't want scar tissue to be able to infiltrate into that tube. So it's important to remember when we talk about that porosity, we're really talking about from the product edge. And, you know, it is semi-translucent, which is a real benefit because the other products on the market, most of them are not translucent. And so it can be very difficult to ensure that the nerve ends are aligned within the conduit, which is something that you really want to do to ensure you don't have fascicular misalignment. So you've heard me say the word extracellular matrix many times throughout this presentation. And so what is an extracellular matrix? Well, an extracellular matrix is composed of the non-cellular components of organisms and tissues. So these are the collagens, the proteoglycans, the glycoproteins, and what they do is provide the structure and support to the cells in the area. And they vary structurally between species and by location in the body. But the really cool thing about extracellular matrices is they can be manufactured into various shapes and forms. And what extracellular matrices do is that, again, they provide that environment that's conducive to cellular infiltration, which then allows the patient's own cells to lay down the new matrix. And that basically remodels that material into a new tissue layer. So you can think of it as that extracellular matrix serving as a blueprint for new tissue. It tells the body where to lay down that new tissue. And in the case of the nerve, that new tissue is an epineurium-like tissue. And so this is what it actually looks like in the clinic. So this is a case that was provided by Dr. Bunke. And this was a case where he used an Axigard nerve wrap around a nerve. And about 6 months later, he had to go into the same area in order to do a tenolysis. And so since he was in the same area working, he took a look at what had happened to the Axigard material that he had implanted 6 months before. And you can see that it formed this nice tissue layer on the nerve, and that he was really very easily able to separate that tissue layer away from the nerve. It doesn't adhere to the nerve at all. It just forms a nice protective layer around that nerve. And so, you know, Axigard as a biomaterial for nerve repair is really, you know, hits all of the things that you want to see in a material used for nerve repair. You know, it's biomechanical, or it's biocompatible. It has mechanical stability, and you know, it just has really nice properties for use around nerve. So now we're going to talk a little bit about the biomaterial properties in comparison to the reconstituted collagens. And so most of the other products that are out there on the market, most of the other conduits are made from reconstituted collagens. And they're either isolated type 1 collagen or isolated type 1 collagen glycosaminoglycan mixtures. And some examples are Neurogen, NeuroWrap, NeuroMatrix. And these conduits are flexible, they're suturable, but they are opaque. They are porous, but their porosity is much smaller than what you see with the SIS material. So you can see here, this is the edge of the Neurogen material in the picture on the bottom. And you can see that that is a a much tighter structure than what we saw in the similar picture that we had of the edge of the SIS conduit. And this porosity allows for, oxygen and nutrients to get into the nerve, but it does not allow the fibroblasts to get in there and start laying down new matrix. So these collagen conduits are degraded by the body instead of being remodeled. And so the reconstituted collagen processing starts by harvesting tendon from a cow. These are typically bovine products. And then it goes through a chemical soak, an enzyme soak, and then it gets homogenized. And then it gets lyophilized, which is essentially freeze drying. And then it gets cross-linked. And so why would they cross-link these conduits? Well, cross-linking does have some benefits. So it provides a really mechanically stable product. And it also allows you to control your degradation rate of your implant. However, it does have some downsides. It can lead to a fibrous connective tissue response. It can actually impede your degradation if you over cross-link. It does impede the cell infiltration into the scaffold, so you don't get that remodeling response. And, in some people, it can cause a foreign body reaction. And so when we look at Axigard versus reconstituted collagen, what we see is that we get much more significant vascular ingrowth into the Axigard material, along with cell infiltration, than you do compared to the processed collagen. And you can see in the top picture, those purple dots again are all cells. And in the Axigard material, you see that they are found throughout the Axigard material, whereas with the collagen, they're lining up along the edge. And that's much more indicative of a degradation response compared to a remodeling response. And then in the bottom picture, the green is the vascular ingrowth. And you see how much more extensive that is in the Axigard material compared to the collagen material. Now, Integra does have a brochure that they have put out specifically addressing the Axigard material. And what they did was they did a rat study, and they used 6 rats total, so 3 rats per group, which is a very small number of animals to use. Typically, in preclinical studies, you see anywhere from 8 to 12 animals per group, sometimes more. And 2 weeks is when they looked at the host tissue response, which is a very, very early time point. What they said is that Axigard elicits a greater inflammatory response than Integra's ultra-pure collagen material. And so, first of all, inflammation is a normal part of tissue remodeling and wound healing. And 2 weeks is, right at the peak of when inflammation occurs in the wound healing response. They also said that peer-reviewed published literature demonstrates that porcine SIS xenografts have been associated with increased inflammation after implantation. And the literature that they use to support this is based on a DePuy implant, which was made from SIS, and it was on the market back in the early 2000s under the brand name Restore. And the problem with this product was that the processing method that they used didn't fully remove the porcine DNA. And so it caused a xenograft response when it was implanted. And so it did cause a lot of inflammation, and the product had to subsequently be removed from the market. But SIS has been studied in over 1,400 published studies, the SIS material that we use, produced by Cook. And so, they've got 1,400 published studies showing that, this material is safe. And so we decided that we were going to replicate the study that they did, and we were going to look at SIS and collagen ourselves. but we used some later time points. And we used a sciatic nerve transection model. And so we found some really interesting things. So we did find that we do have significantly more macrophages that enter our material than the collagen material, which is what Integra showed in their study. But what we did is look at what type of macrophages were present in the material. And what we found is that we had significantly more M2 macrophages, which are the pro-repair phenotype of macrophages, compared to M1 macrophages, which are pro-inflammatory. And when we looked at the Integra material, the Neurogen material, yes, they had significantly less macrophages than we did. But when we look at the type of macrophages that they had significantly more M1 macrophages in their material than they did M2 macrophages. Then we looked at what a stain called alpha smooth muscle actin. And alpha smooth muscle actin can stain two things. One, it's found in the lumen of blood vessels. And two, it can also be found in scar. And so what we found in the SIS material is that it primarily stained the lumen of the vasculature, which you can see in the round profiles that have brown staining in the picture on the top left. But in the picture on the top right, you see the very dense layer of staining, brown staining in the Neurogen material. And what that is indicative of is an encapsulation response. And then the last thing we looked at was axon regeneration, because remember, we use these materials in a sciatic nerve repair model. And what we found is that the SIS material had a significantly more dense axon growth in the center of the conduit and more organized axon growth compared to the collagen material. So really, this study really highlights the importance of biomaterials in nerve repair and the real difference that they can make in how the body responds and how nerves regenerate through those biomaterials. So here are just some more examples of what these things look like. So the top picture on the left is from the initial animal study that we did looking at the Axoguard material around nerve. It was done in a rabbit model because rabbits are known to have a propensity for scarring and inflammation, especially in response to implants. And what we found was that at 6 months, the material had remodeled into this nice new tissue layer around the nerve. It didn't adhere to the nerve at all, and the nerve was able to freely glide within that material. The picture on the right is what this looks like in surgery. So again, this is another case where Dr. Evans had to go back into the same area where he had used Axoguard, and you can see how it forms this new tissue layer around the nerve. And then finally, the bottom left picture is a Neurogen tube, and you can see that it doesn't remodel at all. It just sort of, you can tell exactly where it is, and it just sort of maintains that tubular shape. Now, in the clinical experience, there is a case series that has been published that has looked at the removal of collagen nerve conduits due to unsuccessful implantation. So this was a case series of 4 unsuccessful conduits. And the explant reasons included a foreign body reaction, chronic inflammation, and scar tissue formation. And so figure 3 that you see on the top right is an example of a patient who had a 3-centimeter laceration of the median nerve at the carpal tunnel. And at 6 months, they had no recovery. And when the surgeons went back in, there was so much scar tissue that they ended up having to resect 8 centimeters of nerve and scar. And, you know, they ended up having to repair. Their only option at this point is to repair with an autograft. And so, you know, they went from having a 3-centimeter gap originally to having this big 8-centimeter gap as a result of this inflammatory response induced by this collagen conduit. So we do have some clinical studies that discuss the use of SIS in the clinic. One is from Jordan et al. And in that paper, 71 patients had an Axoguard nerve protector, and there were no cases of infection, persistent inflammation, or recurrent fibrosis. Now, they didn't really have outcomes on these patients, didn't really say whether or not they improved, but with no recurrence of fibrosis or infection or inflammation, it's still an important paper to demonstrate that, in the use of the clinic, we're not really seeing any sorts of issues. And then the second paper here by Papatheodorou, in that paper, they actually looked at outcomes after use of Axoguard. And so these were in patients who had recurrent cubital tunnel syndrome, so patients with a known propensity to scar. And what they found is after use of the Axoguard nerve wrap, there were no complications, and the patients had significant improvements in pain, satisfaction, grip strength, and pinch strength. And 100% of the patients reported improvement.